Good afternoon. So this is the third part of the lab number 4, dealing with enzymes. So in this case, we're going to be dealing with the milk curdling with enzymes. So let's start by defining what is a protease. So a protease is an enzyme that is able to chop other proteins into smaller compounds. And in this case, we're going to be studying the protease bromelain, which is encountered in pineapple juice or in pineapple. So what is milk curdling and why does it happen? So when you have milk, you have normal milk, and let's say you add pineapple juice, in the milk you have these mycel structures which contain casein proteins, and these are in solution, but they are very fragile. And so by adding the pineapple juice, which contains the protease bromelain, this enzyme in the pineapple is able to break this mycel structure and hence causing the clumping of the casein proteins. And that's how you result with this curdling or the coagulation in the milk. And so in this lab, we wanted to study, how does this happen, and what would happen if we say boiled the pineapple juice before adding it to the milk. So these are my materials. I use a pineapple, a blender, 3 cups, milk, containers, cheesecloth, spoons, markers, other spoons to measure, containers, and these are just basically the material that I use. So first I started off by chopping and cutting my pineapple. Then I cut it into smaller pieces. I blended it, and then I put it in a container. Then I separated it into 2 other containers. One was labeled heated, and one was labeled unheated pineapple juice. But prior to that, I used the cheesecloth to drain all the juice so that it was only pure juice. And then one of them, I put it in the microwave and I heated it until it started boiling. And the other, I just left it like that. The next thing that I did is that I labeled 3 other containers. One I labeled milk, the other milk plus juice, and the other milk plus heated juice. And this is basically what the components that it says there is what it would have. So my theory was that by heating the pineapple juice, I know that something will occur to the enzyme. So this is very interesting. So let's see what occurred. And so then I added 1 tablespoon of milk to each one of them, the same. Then I swirled the milk to see if anything would happen, and nothing happened. Then I added 1 tablespoon of the normal pineapple juice, which was not heated, it was just at warm temperature, at room temperature, normal. Then I added 1 tablespoon of heated juice to the 3rd container containing milk. And I waited 5 to 8 minutes to see what would occur. So after the 5-8 minutes, these were my results. The milk remained the same. There was no change. I added nothing to it. It was just the milk. So you could maybe call this my reference or my blank, because I did nothing to it. I didn't change anything to it. Then my milk plus juice, I noticed that there was the formation of these clumps, this curdling. And in my 3rd one, I noticed that there was no change. It remained just like the milk. There was no clumping. The only difference was that was a change in color, but that was just due to the color of the pineapple juice. So why did the milk? So this is what happened to the one with the pineapple juice, the normal pineapple juice. So what happened is that the curdling happened, what I explained before. So since it was normal pineapple juice, nothing was done to it. The clumps formed. As you can see in this image, the walls had little clumps of the casein proteins that the bromelain enzyme, broke down. When it broke down the mycelial structures. And in the heated juice, there was no difference. So what this indicates is that, and this is also my conclusion, this indicates that as we heat an enzyme, for example, a protein like bromelain, it becomes denatured. As by knowledge, we have been taught that enzymes have specific temperatures at which they can work best. And so by boiling it, we denature the protein, the enzyme, and so it's no longer functional, hence it is not able to break down those mycelial structures, and so the casein proteins and the mycelial structure remains intact, and so no clumping occurs. So that's why you can see that in this one there is no change, and in the one where I did the normal juice, the clumping did occur. So that's what happened. Basically, the enzyme became denatured, and so I think that it is very important that enzymes now exist, why they happen in the body. And I know I've been taught before that, for example, when someone has a very high fever, like very high, that this could be the chemistry, the biochemistry behind it, that enzymes cannot work very well under super high amounts of heat, and so this can cause denaturing, and then more other problems in the body and other cascade reactions, or things would not be working well in the brain, in the rest of the body. And so enzymes are imperative in our body, in our human body, and other organisms, of course. Thank you. That was this lab, the third lab experiment. And thank you.